So let's continue our discussion of dominance a little bit and talk about flavors of dominance. Now, we've already seen this type of dominance in the sense of complete dominance. That's that traditional simple Mendelian inheritance. You're either tall or dwarf. You either have Huntington's disease or you don't. And you either have attached or detached earlobes. These are all examples of single gene control and classic complete dominance. Now, a second type of dominance that we will encounter quite a bit is called incomplete dominance. In this situation, the heterozygote exhibits a phenotype that's intermediate to the corresponding homozygous. And these are some examples, and we'll see 4 o'clock flowers quite a bit in talking about this. We'll also, y'all know about sickle cell disease, Tay-Sachs. These are 2 other examples of incomplete dominance, as is hypercholesterolemia. Say that 3 times fast. So I just wanted to touch on really quickly. Tay-Sachs is typically Tay-Sachs disease is a fatal condition. It's a neurodegenerative lysosomal storage disorder, and it's caused by mutations of hex A. Now, in terms of incomplete dominance, what we mean by this, or what's one of what we see in these situations. Let's think about the 4 o'clock flowers in purple color. Here we've got a 2 allele system again, 2 allele system. A big R allele that causes the red flower color, and a W allele that causes the white flower color. Here you've got start out with your P generation, and these are true breeders, meaning they're heterozygous for these alleles. Now we see a red and a white parent. When we get to our F1, what we see here is a pink offspring. So that is not, number one, it's not a trait that you see in the parents. However, it's not either the red or the white, as you would see in the classic complete dominant situation. It's actually a phenotype that's intermediate between these 2. Now, when you actually mate 2 heterozygotes, you actually get a very different ratio of offspring that you would see in a complete dominant situation. Here we still have the classic 1:2:1 genotypic ratio, but in this situation our phenotypic ratio actually mirrors that. So incomplete dominance can generate equivalent genotypic and phenotypic ratios. Now, what's happening at the molecular level? In this case, or in this situation, we're seeing that 50% of that protein that affects the trait is not enough to produce the dominant phenotype. Now, we talked about this a little bit before when we talked about dominance and recessive, but this is the classic explanation. And I want to highlight again this concept of seeing actually 3 classes of phenotypes, which is different from what you would see in the situation of complete dominance, and also a phenotype is not observed in the parent. Now, one of the things is actually kind of cool in terms of an evolutionary sense. Remember, I mentioned previously that sickle cell trait or sickle cell disease is actually a situation of incomplete dominance, or that's where you observe incomplete dominance. So sickle cell trait is actually the heterozygote, or individuals that have sickle cell trait is the heterozygote condition. Overdominance, which is also known as heterozygote advantage, is actually a phenomenon, an evolutionary phenomenon, or a population genetics phenomenon. It's not really a type of dominance, so be careful with thinking that. Really, it's just a situation where the heterozygote is more fit than either of the corresponding homozygous. But this is only in specific situations would a heterozygote be more fit. Typically, overdominance involves autosomal recessive disorders, and in this situation with sickle cell, you've got individuals that if they're a heterozygote or they're affected, i.e. a homozygous for that mutated allele, then they're producing an abnormal form of hemoglobin. So this is actually, I think it's hemoglobin, the beta globin molecule. That's typically the one that's involved with this. So in areas, well, let's just think about it this way: if you're producing a globin molecule that is mutated in some way, the ability of the hemoglobin molecule to hold on to oxygen molecules is going to be compromised. So if you're all of your hemoglobin molecules are compromised, then you are really in trouble. You've got sickle cell disease. So any kind of exertion can cause pretty dramatic symptoms and even quite a bit of pain. In individuals that have sickle cell trait, half of their hemoglobin molecules are the mutant form, and the other half are functionally normally. So these individuals, that's a classic example of incomplete dominance. These individuals are doing relatively okay, except under certain situations. And then, of course, if your globin molecules are not mutated, you're doing fine. Now, in areas where malaria is high in prevalence, you can get a situation where the heterozygote or the sickle trait is actually selected for. So as I mentioned, the two alleles are producing different proteins, and if you want to, you can actually go online, either to OMIM or to some other sites, and you can see that there are over 700 variants of beta hemoglobin alone. So there's quite a few different ways you can have this mutated protein. Now, in terms of the explanation for overdominance, there are three general explanations for it. The one that we're going to focus on is disease resistance, because most of y'all have heard or talked about sickle cell anemia or sickle cell, and so you're familiar with that. So in terms of disease resistance, in this kind of situation, if you, and let's use the example of sickle cell anemia and malaria. So malaria, remember, is caused by Plasmodium, which is a type of protozoan, a parasitic protozoan. Malaria can actually affect a normal homozygote and do very well. So this would be an individual that doesn't exhibit sickle cell. They're doing just fine. All of their red blood cells are carrying oxygen effectively. They're nice big. They're not sickle shaped. So that's the kind of thing that Plasmodium wants. For it to be able to reproduce, it has to have really good cells, or red blood cells. In individuals that are heterozygote, remember they would be sickle cell trait. They have that slightly reduced protein function, and the result is that let's say half of their cells are typical red blood cell shape, and the other half are sickled. If Plasmodium gets into the sickled cells, they can't reproduce. So what this does is generate it's referred to as a resistance to infection, but it definitely doesn't allow that pathogen to propagate. So this is a classic example of overdominance, in the sense that in areas where you have high levels of malaria, it's better for you to be a heterozygote than it is for you to be homozygous and not have any of the symptoms of sickle cell. Now, obviously, if you're homozygous for the mutated allele, then that's got your other problems. Now, two other examples of this are Tay-Sachs and tuberculosis, and then also PKU, and that belief that heterozygotes have a defense against fungal toxins. Now, I want to refer back to this idea of multiple alleles and also with the ABO blood group. So remember that blood type is determined by the type of antigen that's present on the surface of the red blood cells. And these antigens are then in turn recognized by antibodies. So there's actually three different alleles which determine the antigens for the humans. In terms of the, I'm pretty sure most mammals. We'll go with that. So there's allele IA, which encodes or produces antigen A. Then we've got antigen B. And then we've got a situation of a little I allele where you don't get any surface antigen produced. Now, what's happening here is that you've got a situation of codominance. This, in terms of the relationship of the little I allele to both IA and IB, is classic dominance recessiveness. So every time you have IA and little I, that means your blood type is where you're producing the A antigen on your red blood cells, the surface of your red blood cells. The same goes if you're IA and little b. Not you're producing A, but you're going to produce B antigen. Now, however, if you are IA and IB, then both of those antigens are going to be expressed. So this is a situation of codominance. Notice down here, we've got whether you've got antigens or not. Here's a situation where you're homozygous recessive for the little I allele, and then the different situations, whether you're A, B, or A and B. And it's all based on the determining of that antigen on the red blood cells. So this is the classic example of codominance.